Welcome to the deep dive, everyone. As medical microbiologists, I'm sure Bacillus anthracis is a familiar name. But today, we're going to dig a little deeper.
Oh, yeah, we are. Definitely beyond the basics.
We've got MicroWiki, Gemini, even a cool visual guide to help us out. Get ready for some fascinating insights about this bacterium. Even you seasoned pros might be surprised.
It just goes to show even a bug as well-studied as this one can still hold surprises, especially when you consider how it shaped medical history.
Totally. It's like Bacillus anthracis was the OG, the first bacterium we knew could cause disease. Can you imagine the impact of that discovery by Koch and Pasteur?
I mean, talk about a game changer. It basically birthed the whole field of medical microbiology as we know it.
Right. It revolutionized how we thought about disease and how it spreads.
But it's not just history, right? Bacillus anthracis is still a major player today, sadly because of its potential as a bioweapon.
Oh, absolutely. That adds a whole other layer of complexity to studying it. I mean, the 2001 anthrax scare in the U.S., that really drove home the biosecurity concerns.
For sure. So let's dive into what makes this bacterium so unique, eh? Let's start with those virulence factors.
Yeah, It's crazy how Bacillus anthracis has those two plasmids, PX01 and PX02.
Like little blueprints for its weapons?
Literally. I was reading in MicroWiki, the anthrax toxin is actually a 3-part protein system.
Yep, 3 parts. It's a pretty sophisticated mechanism. You've got the lethal factor, edema factor, EF, and the protective antigen, or PA for short. Each one has a specific job to do, disrupting the host's cells.
Okay, so walk me through this. The protective antigen, that's the key to getting the other 2 factors into the host cell, right? Like a Trojan horse.
You got it. PA binds to receptors on the host cell, then sneaks LF and EF right in.
Sneaky little buggers.
Oh, yeah. And once they're inside, LF goes to work. It acts as a proteus, messing up critical signaling pathways that cells need to survive and for the immune system to function.
Sabotaging the cell from within. Clever, but deadly.
Exactly. And then there's EF.
What does that one do?
EF is an adenylate cyclase, so it cranks up the levels of cyclic AMP, or CAMP, inside the host cell, throws the whole signaling system out of whack.
Leading to?
Edema. Big time. That's the fluid buildup you often see in anthrax infections.
Makes sense. So these 3 proteins, they work together, causing all this damage. Okay, now what about that protective capsule, the one encoded by PX02?
This is where things get really interesting.
Right. It's not your typical capsule, is it?
Nope, not at all. Most bacterial capsules are made of sugars, polysaccharides, but this one's made of D-glutamate, amino acids.
Whoa, that's wild. A total evolutionary curveball.
It makes you wonder, wouldn't a regular sugar capsule be better in some environments? What are the trade-offs,
Right. Why go this route?
Excellent question. It really highlights the back and forth, the constant arms race between pathogens and the hosts they're trying to infect.
So what advantage does this D-glutamate capsule give Bacillus anthracis?
Well, it's all about stealth. It helps the bacterium dodge the host's immune system, resists phagocytosis, meaning those immune cells can't just gobble it up.
So it's like a cloaking device, allows it to slip past the defenses. I bet that's a big part of how it causes disease.
Huge. Evading phagocytosis means Bacillus anthracis can multiply like crazy, spread throughout the host, makes the infection much worse.
Okay, so we've got this toxin and this super sneaky capsule, but how does it actually get into the host to begin with?
That's where its life cycle gets really interesting, and in particular, its ability to form those endospores.
Ah, yes, the infamous endospores. They're tough little buggers, aren't they?
Incredibly tough. Basically, when things get rough, nutrients run low, temperatures get extreme, Bacillus anthracis says nope to its normal state and forms these incredibly resistant endospores. Think of them like little time capsules.
So they basically hibernate, just waiting for the right conditions to come back to life.
Precisely. And they can wait for a long time. We're talking years, even decades. Heat, cold, dryness, even some disinfectants, they don't care.
Talk about a survivalist. But how do they actually cause infection then?
Simple. Those dormant spores just hang out, waiting. Then, when they come into contact with a potential host, bam, they germinate. Back to their active, vegetative, bacterial form.
Usually 3.
Break in the skin, inhalation, sometimes even ingestion.
And then the cycle starts all over again. It's remarkable, this ability to survive anything, then jump right back into action when the opportunity strikes.
It's a testament to evolution, really. It's been shaped over millennia to be a master of survival and transmission.
Now, thinking about how it looks under the microscope, our visual guide shows these large colonies, kind of matte, almost a ground glass look.
Yeah, and it's important to note they're not hemolytic, no pigment either. That helps us tell them apart from some other bacillus species.
Good to know. So what about the individual cells? What do we see there?
Large gram-positive rods, often in chains, and remember those unique cell wall features we talked about?
No teichoic acids, those unglycosylated S-layer proteins?
Exactly. Subtle but crucial for making a proper ID.
It's amazing how much those tiny details matter when you're trying to tell these closely related species apart.
In the world of medical microbiology, they can make all the difference. For accurate diagnosis, for effective treatment, they're critical.
So we know Bacillus anthracis is a facultative anaerobe, meaning it can grow with or without oxygen. And our sources say it grows well on both aerobic and anaerobic media.
Versatile little guy, isn't it? That's part of what makes it so successful. It can thrive in a wide range of environments.
Speaking of versatility, I was curious about its motility. I read it has a flagellin gene, but it's non-modal. Like, it has the gene, but mutations mean it doesn't move. Why would it lose that ability over time?
The mysteries of evolution. It's hard to say for sure. Maybe motility wasn't so helpful in its usual environment.
So it just ditched it.
Could be. Maybe its ability to form spores and hang out for ages was a better survival strategy. Who needs to move when you can wait it out, right?
Makes sense. Streamlining the toolkit, focusing on what works best.
Exactly. Even simple organisms are constantly evolving, adapting to their surroundings. It's fascinating, really.
It is. Now let's shift gears a bit. As medical microbiologists, the clinical side of things is super important.
Absolutely. Understanding the different type of anthrax, how they present, that's key for diagnosis and treatment.
So let's talk cutaneous anthrax first, the most common type, usually from contact with infected animals or their products. What should we be looking for?
Starts with a small bump, a papule at the entry point, painless. But then it develops into this black eschar, a dark necrotic lesion. Nasty stuff.
And usually there's swelling around it, maybe fever, general feeling of being unwell.
Right, the whole package. But the good news is, cutaneous anthrax, it's usually treatable with antibiotics, especially if caught early.
Catch it early, that's key. Okay, now pulmonary anthrax, that's a different beast.
Very different.
From inhaling spores, right?
Yep, and when those spores get into the lungs, they germinate, release their toxins. It's a rapid, serious infection.
Much more serious.
Oh, definitely. You'll see fever, cough, chest pain, trouble breathing. Bad news all around.
And if it's not treated?
It can be fatal, unfortunately. Those toxins cause major damage, not just to the lungs, but other organs too. Respiratory failure, septic shock. It's not good.
Definitely not good. Okay, lastly, gastrointestinal anthrax. The rarest form, right? From eating contaminated meat?
Right. This one can present with a lot of different symptoms. Nausea, vomiting, abdominal pain, bloody diarrhea is a mixed bag.
I imagine that makes diagnosis tough. It could look like a lot of other GI issues.
You're right, it does. It takes a high index of suspicion to catch it, especially since it's so rare.
Okay, so we've covered the main types of infection. Let's move on to some of the newer research, especially anything that might have clinical implications. I was reading in Gemini about this interesting detail regarding anthrax protective antigen, or PA. It seems it can actually cause lysis in human cells, basically making them burst.
Yeah, it's a bit of a head-scratcher. Why would a toxin that needs host cells want to destroy them, right?
Right. So what's the deal? How does it work?
Well, it turns out PA interacts with this receptor on human cells called TEM8.
Okay, and?
TEM8 has a low affinity for PA, meaning the bond between them isn't that strong.
So PA can attach, but it also detaches pretty easily.
Exactly, and that detaching is important. Once PA comes off TEM8, it can insert itself into the cell membrane, creating pores, and those pores, they cause cell lysis.
So TEM8 is like a temporary docking station, then PA goes and punches holes in the membrane. Smart, in a destructive kind of way. But then why would it do the same with the CMG2 receptor? That one has a high affinity for PA, right?
Excellent question. And the answer is pretty cool. CMG2 binds PA so tightly it doesn't let go. So PA can't insert into the membrane and cause lysis.
It's like a key that fits the lock perfectly, but you can't turn it.
A perfect analogy. This just goes to show how intricate these toxin receptor interactions are. Even a tiny difference in how strongly they bind can completely change the outcome.
Definitely highlights the importance of research. We need to keep digging to fully understand how Bacillus anthracis causes disease.
Absolutely. The more we know, the better we can develop effective treatments, prevention strategies, the whole 9 yards.
Okay, I think we've covered a ton of ground here, from the history of Bacillus anthracis to its virulence factors, how it infects people, the whole shebang.
And we even touched on some cutting-edge research. Always exciting to see it's new.
But before we wrap up, I want to test your knowledge a little bit. Exam-style questions. Are you ready for the challenge?
Hit me with your best shot. Let's do this.
Okay, question number 1. What's that key feature of Bacillus anthracis that lets it survive for so long in a really harsh environment? And how does that help it cause infection?
You're talking about those endospores.
Bingo.
Those things are tough, man. They let the bacteria just chill out, lie dormant when conditions are bad, stuff that would kill regular bacteria. Then, when a host comes along, boom, those spores germinate, start causing trouble.
It's like a bacterial sleeper cell. Okay, question 2. You're in the lab. You get a sample from a patient, suspected cutaneous anthrax. What are you looking for to confirm the diagnosis? What are the key lab findings?
All right, first things first, gram stain. Got to see those large gram-positive rods, often in chains. It's classic anthrax. Then culture that sample, see what grows. Bacillus anthracis has some pretty distinctive colonies.
What do they look like?
Non-hemolytic, no pigment, and they've got this, I don't know how to describe it, a ground glass appearance. It's kind of unique.
So even how the colonies look could be a clue. What else?
The big one, you got to confirm those virulence plasmids are there. PX01 and PX02, those are the calling card of Bacillus anthracis. They're what code for the anthrax toxin and that protective capsule.
Right, those plasmids are the smoking gun. Okay, question 3. Let's talk about how anthrax toxin actually works. Break down the mechanism for me. How do those 3 components team up to cause all that damage?
Okay, so picture this, the protective antigen, PA, it's like the key. It binds to receptors on the host cell surface, opens the door, so to speak.
For the other 2 components.
Exactly. Lethal factor, LF, and edema factor, EF. They hitch a ride with PA, get right inside the cell.
Sneaky. So PA is like the delivery truck.
Perfect analogy. Now, once inside, LF gets busy. It's a protease, so it starts chopping up key proteins involved in, well, everything. Cell survival, immune responses, you name it.
Talk about a targeted attack. What about EF? What's its role in all this?
EF's job is to mess with signaling. It's an adenylate cyclase, which means it ramped up cyclic AMP levels inside the cell.
And that causes
edema, fluid buildup. So you've got LF shutting down essential cell functions and EF causing all the swelling, a 1-2 punch that really throws the body out of whack.
A very effective, albeit nasty, strategy. Okay, question 4. We talked about the Bacillus anthracis capsule earlier. What makes it so special compared to other bacterial capsules? And what's the advantage of that?
So most bacterial capsules are made of sugar, polysaccharides, but Bacillus anthracis, nope. Its capsule is poly-D-glutamic acid.
Meaning?
It's made of amino acids. It's a protein capsule, not a sugar capsule. That's pretty unusual.
Huh. Interesting. But why go that route?
Camouflage. That unique composition helps Bacillus anthracis fly under the immune system's radar. It's harder for immune cells to recognize it so they don't attack as effectively.
So it's like a disguise. Sneaky little bugger. Okay, last question. A patient comes in, fever, cough, chest pain. They've been working with imported animal hides. Thinking about anthrax, what's the most likely diagnosis here, and why is it so worrisome?
Sounds like pulmonary anthrax to me. Inhaling those spores from contaminated animal products, that's the classic route.
And why is this type so dangerous?
Because it's often the most severe. Those spores get into the lungs, start germinating, releasing toxins. Things can go south very quickly.
So it's a race against time.
You got it. Early diagnosis, aggressive treatment with antibiotics, those are crucial. That's the best shot at survival.
True, those are some tough questions. I think we survived our microbial interrogation, though.
We did. All right. Covered a lot of ground today. Really got into the nitty-gritty of Bacillus anthracis.
From its history to its unique feature, those tricky diagnostic challenges, everything. But before we call it a day, there's one more thing I wanted to touch on, something I found pretty interesting in our research. It really shows you how complex that interaction between microbes and our immune systems is. Even the smallest differences, like how strongly a toxin binds to a receptor, can have huge consequences.
Absolutely. And it's a great reminder for us in microbiology that the details matter. Sometimes those little things hold the key to understanding the bigger picture of how diseases work.
And speaking of the bigger picture, we talked about Bacillus anthracis as a potential bioweapon. It's a scary thought, but a real one, unfortunately.
Yeah, no doubt. It's a reminder that scientific knowledge can be powerful, but it can also be dangerous if it falls into the wrong hands.
Absolutely. The 2001 anthrax attacks in the U.S. really brought those biosecurity concerns to light. Made us all realize how important it is to be prepared, both in terms of public health and making sure scientific knowledge is used responsibly.
Couldn't agree more. As scientists, medical professionals, we have a responsibility to be aware of these threats, do everything we can to prevent them. It's a serious issue.
Definitely. Now, before we wrap up our deep dive here, there was this one thing I found kind of intriguing in our research. MicroWiki mentioned that Bacillus anthracis actually has a gene for flagellin.
Flagellin, you mean the protein that makes up those bacterial flagella?
Yep, that's the one. But we know Bacillus anthracis isn't motile. It doesn't move. So why would it have that gene if it doesn't use it? It's like having a car but no engine.
Huh, that's a great analogy. It is a bit of an evolutionary puzzle, isn't it? I mean, there are a few possible explanations. Maybe Bacillus anthracis had motile ancestors but lost the need for movement over time.
So it's like those cave creatures that lose their eyesight because they live in darkness. If you don't use it, you lose it.
Exactly. Evolution in action. Or maybe that flagellin gene has a different role in Bacillus anthracis that we haven't figured out yet.
So it could be doing something totally unrelated to movement. Fascinating.
Who knows? It just goes to show there's still so much we don't know about these microscopic guys. Even genes that seem useless might have a hidden function, we just haven't uncovered it yet.
Well, I think that's a perfect note to end on. Even with all we've learned about Bacillus anthracis, there's always more to uncover. It's a world of endless possibilities.
And that's the beauty of science, right? Always more to explore, more to learn.
Absolutely. Well, thanks for joining me on this deep dive into the fascinating world of Bacillus anthracis. We've covered a lot of ground today, from its history and those unique virulence factors to the ins and outs of diagnosing and treating anthrax infections. Hopefully, you've all picked up some valuable knowledge, maybe even a newfound appreciation for this tiny but powerful bacterium. And as always, keep those curious minds engaged. There's a whole microscopic universe out there just waiting to be discovered.